the metaphase, the nuclear membrane completely disintegrates and the chromosomes are spread out in the cytoplasm at the beginning of metaphase.
Yeah, so metaphase basically starts from the nuclear membrane completely vanishing and all the chromosomes coming out and spreading. Okay, then from the spreaded form they start aligning at the center. Yes, continue.
The metaphase is characterized by all the chromosomes coming to the center of the centromere with one chromatid attached to spindle fiber from one pole and its sister chromatid attached to the spindle fibers from another plate. This is called metaphase plate.
Yes, and then the next phase is anaphase.
Yes, anaphase. At the beginning of anaphase, the chromosomes that were arranged at the metaphase plate begins to split apart, and from here we no longer call them sister chromatids. They are known as daughter chromatids. The daughter chromatids is separate. The daughter chromatids is daughter chromosomes, which is also called daughter nuclei.
Daughter chromatids form the daughter chromosome. You know, chromatids are not condensed, then they become condensed to form chromosome, and then when the nuclear membrane comes around, so the same daughter chromatid forms the daughter chromosome and then the daughter nuclei. These are the terms in succession that are used. Okay. Yes.
The daughter chromatids begin their journey towards the opposite poles. This is karyokinesis.
Yes.
Then we have telophase. In telophase, chromosome clusters reach their respective poles and begins to decondense, which means their individual identity is lost.
Yeah, so you cannot see them any longer as chromosomes or chromatin threads. No, they are not visible. Yeah.
Then the nuclear envelope forms back around the chromosome at each pole to form two daughter nuclei in one cell. And
Other cell organelles, which was the endomembrane system, they also appear.
Begin to form and the initiation of cell division.
Yes, right. So till telophase, we have finished the karyokinesis. So telophase ends with karyokinesis done. Now what happens?
Then the next stage is cytokinesis, which means the division of cytoplasm. In animal cells, cell division is achieved by forming a furrow in the plasma membrane. This furrow gradually deepens in the cytoplasm and ultimately joins in the center to divide the cell into two parts, outside and outside in manner.
We discussed that this is outside in manner in animal cells, whereas in plant cells it is different, right?
Yes.
What happens in plant cells?
In plant cells, the cell wall in plants is relatively inextensible. Therefore, they undergo cytokinesis in an inside-out manner. The wall formation starts in the center of the cell and goes outwards to join the existing lateral walls, and this cell wall formation begins to form a cell plate.
Yes. Right.
Then we discussed about the multinucleated cells.
And what is that?
In some organisms or cells where karyokinesis is not followed by cytokinesis, give rise to a multinucleated condition, which is a syncytium.
Syncytium. It's called a syncytium.
Syncytium. Example: liquid Edukiran in coconut or coconut water.
So basically, coconut water is a syncytium of just nucleus present, free nucleus. Remember, I told you that we are just drinking the free nucleus.
Yes.
Okay.
We discussed a question. Significance of meiosis.
Your voice is a little lagging to me, Asha. Yeah, would you like to come closer to the mic? I think that will be good. Yeah.
Am I audible now?
Yes.
Then we discussed the question, significance of mitosis. In animals, only diploid cells undergo mitosis. In plants or insects, haploid or diploid cells both can undergo mitosis.
Yes.
The genetic component of daughter cells remains identical or same as the mother cell. Growth of cell repair.
Not audible to me. Okay, now you're audible again.
Can you hear me now?
Yes. Can you please repeat the last line again? Just the last line.
The genetic component of daughter cells remains identical or same as the mother cell.
Yes.
Then the next one is growth, cell repair or tissue repair, replacement of cells. The continuous growth of plants throughout their life is due to mitotic divisions in their meristematic tissues.
So in this meristematic tissues also mitosis happens. So these are all the significance of mitosis, and then we started with meiosis. So let me come to Iram from here. Iram, would you like to just tell what we discussed about meiosis, and then we will start with.
Sure.
Yeah.
Okay, in meiosis, it is a process where a single diploid cell undergoes cell division twice to form 4 haploid cells. These are gametes, sperms in males and ovumes in females. There are two meiosis. There's Meiosis-1 and Meiosis-2. Meiosis-1
yes, there are 2 cycles in one meiosis. So it is like you can say there are 2 cycles of cell division in one complete cycle of meiosis. Okay. Saying that there are 2 meiosis puts it like that there are 2 different types of meiosis. Okay,
Yes, sir.
Yeah.
Meiosis-1 is reductional division, whereas Meiosis-2 is equational division.
Yeah, and we realize that why meiosis is important, because to maintain the ploidy constant over generations, all the gametes have to be formed through meiosis and not mitosis, right? And then I gave you this homework to go back and read the 3 pages to finish meiosis up. So, sorry. Yes, you can see the screen, right? So you have made your notes, right, of meiosis. Let's start with the characteristic features of meiosis. Iram, what are the characteristic features of meiosis? And this is also the question. On the first page, you must have studied these 4 bullet points in your NCERT, right?
Yes, sir.
Yes. So what are these?
It brings genetic variation.
Yes, that's one. But okay, let me go step by step. So let me first introduce it to you and Aisha both, then we'll go with your notes, okay? So pay attention here, Aisha. So meiosis, first thing is, the feature is that it involves 2 sequential cycles of nuclear and cell division. Okay, so write down It involves two sequential cycles of nuclear and cell division. Nuclear division and cell division, which is known as Meiosis-1 and Meiosis-2. Correct? This is clear? But it has only one cycle of DNA replication. You understand? Now, where does it start? That is the second characteristic feature. So Meiosis-1 is, it begins after the parental chromosomes, they replicate to produce identical sister chromatids at S phase. You know, sister chromatids are formed at S phase. So Meiosis-1 begins after the parent chromosome replicates. And forms identical sister chromatids at S phase. So here also the interphase is there. So interphase is G1, S and G2. Then starts the M phase. Now M phase can be for mitosis or it can be for meiosis. But in that M phase, now it is longer for meiosis, because instead of just having one cycle, prophase, metaphase, anaphase, telophase, and then cytokinesis, there are two cycles. Prophase, metaphase, anaphase, telophase, first cytokinesis, then again prophase, metaphase, anaphase, telophase 2 with second cytokinesis. So the M phase is double. Clear, right? The distinction is clear. But interphase is same. The G1, S and G2 will be same as of after every division. It will be same for every cell as of mitosis. Is it clear? Now, one important characteristic feature of meiosis, which is different from mitosis, I'm making a star mark here, is that you told me, Iram just told me that meiosis produces variations, right? There's a variation in genes. What do you mean by variation, Iram? When you said that meiosis produces variations, what do you mean by that? Or what do you understand by that sentence? Because if you understand that, then you won't have to memorize it. Iram, are you there?
Yes, sir.
Yes. So how do you think variation? What are variations basically? Let's discuss this a little bit. So let's say there is this chromosome number 1 from the father, and the same chromosome number 1 from the mother. So this is from the mother and this is from the father. So we have one copy from the mother, one copy from the father in each cell. That is why it is called 2n, right? Now, this is just one chromosome which has not replicated yet. So tell me, these chromatids, are they sister chromatids or non-sister chromatids?
Non-sister.
Yes, they are non-sister chromatids. Do you understand why, Iram?
Yeah.
Yeah, because the sister chromatids will be formed in the S phase, where both the chromosomes will just replicate in DNA. Understand? So these two are sister chromatids. Is it clear?
Yes.
Now, in the non-sister chromatids, or in this, because they are identical, let's say this green area talks about a gene. So because they both are identical, so there is a gene present here. In both these chromosomes, because they are the same chromosome coming from two different parents, they are called homologous chromosomes. What are they called? Homologous chromosomes. Homologous chromosomes basically have the same number of genes, the same genes at the same location. So these chromosomes are basically same. They have the same length, same P and Q arm, same position of the centromere, and all the same genes in the same orientation. But what is different is what gene, what is the type of gene or the trait it can carry, allele of the gene. So for example, let's say this gene, green, one marked here, is for eye color. So mother can have a blue eye color, right? Or let's say brown eye color. Okay. And father had blue eye color. The gene is for eye color, you understand? The gene is for eye color, but the alleles, which means the alleles are
So that one dominant and recessive?
exactly. So alleles are, one has to be dominant, other has to be a recessive, that thing. So you have studied that already? I was thinking, if not, then how should I explain
Yeah,
it to you?
10th.
Yes.
Yeah, you have studied it, right? Okay, very good. So you already know that alleles are the two different forms of the same gene. So gene here is for eye color. Now, if there is some change in this gene, it will give brown color to the eye, and the other will give blue color to the eye. So these two are called alleles. So two homologous chromosomes can have two different alleles. That is the difference. Yes. And let's say there is another gene here on this chromosome, which I have marked in red. And this is for hair. So let's say the father has brown eye color with black hair. And the mother has blue eye color with, let's say, blonde hair. Okay. So this is the parental combination, brown and black, brown eyes, black hair, blue eyes, blonde hair. This is the parental combination. One parent was having blue eyes with blonde hair. The other parent was having brown eyes with black hair. Is it clear till here, both of you?
Yes.
Now tell me, if they have kids, let's say, what are all the possibilities that can be present in kids? Kids, what could be all the possibilities in kids? First is, either the kid has the parental types. The kid has brown
Black hair.
with black hair. So if this is the parental, if this is progeny one, first kid, then you will say that brown and black both were dominant from the father. So mother's gene did not express in the kid, right? You will say
Yes.
that this kid has, like, this kid is like his father. That's how we say, no? Have you seen when kids are born, we say that, okay, this kid looks like the father, this looks like the mother. Sometimes people go one step ahead, it looks like the grandfather. The nose is like the grandfather, the chin is like the father, like that. So basically, all these are characteristic. The other possibility is the kid can have blue eyes
with blonde hair.
with blonde hair. Now, in these two possibilities, in these two combinations, do you see anything different from the parents? No. Both of these are parental combinations. Yes or no? No variation. But sometimes it is possible that you will see a kid with brown eyes but blonde hair. This is possible?
Yes.
Yes.
Now, brown was a characteristic of the father, but blonde hair came from mother. So for one gene, it expressed this, and for the other gene, it expressed this. How? Or let's say, this is also possible that it has blue eyes with black hair. So these two, do you see they are non-parental combinations? Can you understand?
Yes.
And this is actually what happens in nature in reality. We are always combinations of our parents. You will say that, unlike in some cases where, either one kid has gone, one kid looks exactly like the father and the other kid looks exactly or very much like the mother externally. But mostly kids have mixed characteristics. The eyes will be like the mother, or the eye color will be like the mother, but the hair texture can be like the father. They will have nose like the father and chin like the mother. So mixed features. You understand? Those are called non-parental combinations, or in other words, they are called recombinants. Recombinants or variations. So this combination, brown and blonde, none of the parents had. Yes? Yes or no? None of the parents
Yes.
were brown with blonde, and none of the parents were blue with black, blue eyes with black hair. So these two are variations from the genes that were expressed in the parents. You understand that, everyone? So how does this happen? Okay, this happens through meiosis, because during meiosis there is an event that is not explained very well in your book, but I will explain it so that it will help you in class 12th as well. So wherever you go to study class 12th from, remember this concept. This concept, before I explain the third point, it is called crossing over during meiosis. It is a literally physical crossing over happening between the non-sister chromatids of homologous chromosomes. So write down this one point first. Meiosis involves pairing of homologous chromosomes. You understood homologous chromosomes? The same chromosomes coming from two different parents is homologous chromosomes, carrying the same genes, but it may carry different alleles. So meiosis involves, yes,
Thus,
Hiram?
homologous chromosomes are 23 in pair.
Yeah, we have 23 pairs. Homologous chromosomes are 23 pairs in females, but in males, 22 autosomes are homologous because the sex chromosome is X and Y. So it is not the same chromosome coming from two sources. So it is actually a good question you asked. So if you see in males, there are only 22 pairs of homologous chromosomes in a diploid cell, right? Because the 23rd pair is not a homologous chromosome. It is two different chromosomes, X paired with Y. Whereas in females, all 23 pairs are homologous chromosomes because they are homogametic. They have X and X. You understand, Hiram?
Yes.
Yeah. So write down. Meiosis involves pairing of homologous chromosomes, and there is a recombination between, and there is a recombination between non-sister chromatids of homologous chromosomes. There is a recombination between non-sister chromatids of homologous chromosomes. First, make this diagram, this one, and mark all these things first. This is chromosome number 1. This is also chromosome number 1. Make this diagram, mark everything, and write this and let me know. Done?
Yes.
Okay. Now, let's come here. So this crossing over during meiosis happens between the non-sister chromatids. Now let me use 2 colors. So let's say this is one chromosome. Okay. And this is another chromosome. I'm just using 2 different colors, but just know that both of them are homologous. Okay. This is also chromosome number 1, let's say, and this is also chromosome number 1. So these 2 are homologous, correct? Understood?
Yes.
Now, these homologous chromosomes will form sister chromatids during DNA replication. I'm using these 2 different colors for a reason. I'll tell you why. Okay, so during DNA replication, let's say both the homologous chromosomes have made their sister chromatids. So if you see, these 2 are now sister chromatids, and these 2 are also sister chromatids. Correct? But these 2, they are non-sister chromatids. You understand? Can you see the screen, both of you? Are you understanding?
Yes.
Good. Now what happens is, let's say it contains a gene, a red gene here. Okay. So same will be here also. The same gene will be here also and here also, because these are homologous chromosomes. But the difference will be that, let's say this gene is for height and one chromosome coming from the mother contains capital T as the allele for dominant height, right? Tall height. Capital T is for tall. This you have studied in class 10th.
Yes.
Yes.
And this contains small t here. But the gene at the same place is for height only in both the chromosomes. One is having a allele capital T, another is having a allele small t. Now, let's say there is another gene present here, which is yellow. This yellow. Okay. What gene you would like this to be? Tell me. Any gene that you have studied from class 10th. Color of the seeds, round and yellow and green, right?
Yes.
So let's say this contains capital Y for yellow. So for green, what should I write?
Small y.
Small
Yeah.
y.
Right. We will not write G. It will be small y. And then let's say there is one more gene. Which color should I use now? Let me use gray. Here. Is gray visible to you?
Yeah.
Yes.
Yeah. And this is for what? Tell me another one more gene.
Round and wrinkled.
Yes. So capital R, round, and this is small r for wrinkled. So clear, we are studying 3 genes together. So this is during meiosis, what happens is these 2 chromosomes literally come close together. This is our first chromosome. And this is our second chromosome. And they literally make this kind of a structure, which is called crossing over. Do you see this structure here?
Yes.
This is called chiasmata.
Smarter.
Chiasmata, or this is the actual event of crossing over of chromosomes. Physically, they are crossing over. Now let's again make the position of the genes. So T, 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 T. Capital T, small t. Small t, yes. Second was yellow gene here. This was capital Y, small y and small y. Clear? And the third was gray here. Now pay attention. This was here also there is a gray and a gray. Here also there is a gray. If I write, it will be capital R. And this blue here is capital R, small r, and small r. So you see that R, both the R's, the gene R is getting crossed over with each other.
Yes.
As a result, what happens is this. When this crossing over is over, so physically what happens, the chromosome breaks from the region, from the point they are crossing over with each other, they break and get joined to adjacent chromosomes. As a result, this chromosome will have a green region with it. Understand? And the green chromosome will have a. You understand how it happened?
Yes.
This got broke and joined to it. Basically, exchange of genetic material happened. So at this level, What is happening? So at this level, during the chiasmata, there is exchange of a part of chromosome between homologous chromosomes, non-sister chromatids. You understand, everyone? This is called recombination. So now if I make the gene, let's make the gene. Same location the genes will be present. This is capital T, small t, small t. Tell me. Then there will be a yellow one.
Capital Y,
Yes, capital Y.
capital Y, small y.
Small y. Very good. Then let's go.
Capital R, small r.
Let's go to this gene here. So this is the gray one, this is the gray one, this is the gray one, and this is the gray one. Yes, tell me.
Capital
This will be
small r.
This will be
Small r.
Small r.
Yes, capital R, small
Small
r.
r.
Yes. Now look at these 4 chromatids. At meiosis, what happens is, how does meiosis forms? What is the end product of meiosis? From your notes, tell us. You know, what happens at the end of meiosis? How many cells are formed?
4.
From one diploid cell, 4 haploid cells are formed. Haploid means they will have only one copy, right? So to have one copy, the cells need to divide all these 4 chromatids. This is chromatid 1, this is chromatid 2, this is chromatid 3, and this is chromatid 4. They all will separate from each other, right? And one chromatid will go into each cell. Now this chromatid 1 will have the gene sequence as capital T, capital Y, and capital R, right? Is this like the parent? Yes. The second one will have capital T, capital Y, small r. Is this like the parent or is it a recombinant?
Recombinant.
Yes. The third one will have small t, small y, and capital R. Parent or recombinant?
Recombinant.
And this is small t, small y, small r. Parent or recombinant?
Parent.
Parental. So you see that 1 and 4 are parental types, which is called non-recombinants. No recombination is there. But if you see these two, they are recombinants. Or I should make it here for more clarity. If you look at these two, they are recombinants, or I can say non-parental type. Is it clear? This is how during meiosis, when actually from one cell having a diploid genome, okay, so can you see that DNA replication happened just once? That is here. So we started with one single line, right? One chromatid of each. Then DNA replication happened and they became 2. Total 4 chromatids, 2 sisters in each chromosome. Yes. After that, we did not do any DNA replication. We just crossed it over, and in the next cycle, when everything got separated, we can form 4 haploid cells, 2 of which will be recombinants and 2 of which will be non-recombinants. this is how variations comes through meiosis, through this process of recombination, which is known as crossing over. Is it clear?
Yes, sir.
Both of you? Clear?
Yes.
This is not explained, but this is very important a concept for you. So please, it will help you in entrance and everything. So please draw this diagram clearly and let me know after you're done. Can you see it on the screen?
Yes, sir.
Draw and mark everything. This is recombination that happens during meiosis. Then we'll go to meiosis. Make it clear. Take some time. Don't worry. Done, Aisha Neera?
No, sir.
Yes, no?
No, sir.
Yes, Iram?
Yeah, done.
Done? Okay. So you understood how variations are produced? You have written the third point, which I have said, that meiosis involves pairing of homologous chromosomes, right? You've written the third point? Yes or no? If not, then write down meiosis involves pairing of homologous chromosomes and recombination occurs between recombination happens non-sister chromatids. Okay. And the last thing about meiosis, the last feature is that from one diploid cell, 4 haploid cells four haploid cells are produced or are formed at the end of meiosis. Meiosis-2. Meiosis-2 is where it ends. And you already know that Meiosis-1 is reductional, but Meiosis-2 just mimics the mitosis. Yes or no?
Yes.
Yes.
So, I had a question.
Yes, Iram.
So, how long is meiosis?
Meiosis, it depends on in which system and for what purpose it is being done. So mostly it's for gamete formation and not, but in some systems meiosis also happens for like spore formation, you know. For example, in from after this we will come to chapter 1 and 2. There you will read slime molds and fungi, so they make spores. So every cell cycle, mitosis also, as I told you, mitosis is different. Remember, in E. coli it was just 20 minutes. Cell can divide in 20 minutes. For yeast it was 90 minutes. For humans it was 1 day, 24 hours, right? Same is with meiosis. It depends in which system it is happening and for what purpose it is happening. But it is longer than mitosis. You understood? Because there are 2 cycles of nuclear and cell division. Is it clear, Iram? Does it answer your question?
Yes.
Yeah, so there's no fixed thing as such. Okay, now coming to Iram. So you have made the notes, right? So let's start meiosis now. So Meiosis-1, let's start with M1. What are the stages in Meiosis-1? First is
Prophase 1.
prophase
Metaphase 1.
1. Okay. Then metaphase 1.
Anaphase 1.
Anaphase 1.
Telophase 1.
It is telophase 1.
Cytokinesis 1.
And then finally it is cytokinesis 1. Everything looks same like meiosis, but the trick is here. Prophase is the longest phase, right?
Yes.
Longest and the most complex phase of meiotic division cycle, especially the first meiotic, because prophase 1 itself is divided into 5 components. First is, you have made these notes, right? It's called leptotene.
Yes. Leptotene, zygotene.
Yes, in one line, what happens in lactoprotein?
Beaded structure. Chromosomes appear like beaded structure.
Yes, so which means compaction happens, right? Compaction of chromosomes. What she has used the term beaded structure. Okay, compaction. Then the second thing is, what is the second thing? Zygote.
Zygote.
Okay, and what in one line, what happens in zygote?
Chromosomes will start to pair,
Yes,
and this is called as synapses.
chromosomal pairing begins. And this is called
Synapses.
synapses. Perfect. Then meiosis third. Sorry, what is the
Packaging.
third in prophase 1? Pachytene. Tell me in one line what happens in pachytene.
Crossing over between two sister chromatids.
Yes, so but in this stage the 4 chromatids become visible, and what happens? Crossing over. So what I have told you above, can you see where it comes?
Yeah, packaging.
Crossing over between two non-sister chromatids. Okay, leading to recombination. You understand? This is the most important phase of prophase 1, pachytene prophase 1, which causes all this thing which you have made, right? This is pachytene basically, where the recombination happens. Then fourth,
Diploide.
yes. What do we mean by diploide in one line? The chiasmata, which I showed you, starts
It will be
separating.
visible.
It will be visible and it will start the X-shaped structure, which is called the chiasmata. It actually will start to separate. Okay, only the chiasmata is visible. Chiasmata is visible. This was chiasmata. Remember I told you this part? So the whole chromosome will separate from each other, but just the chiasmata will be visible. Okay, you can also say dissolution of synapses. Remember this synapses formed in zygotine, so that in pachytene crossing over can happen, and then in diploide this synapses is separated. Only chiasmata is visible. Is it clear, everyone? That's why I explained you this thing, because NCERT does a very bad job here. It doesn't tells you about what is crossing over, but tells you about all this theory. It becomes difficult to memorize or visualize in your brain, right? Now I think it must be easier, especially for Iram, because she has already made her notes. Is it getting easier for you to understand, Iram? Yep, cool. Then what is the last?
Diakinesis.
Diakinesis. What is diakinesis?
It's the terminalization of chiasmata.
Yes. Can you see that the word kinesis is here? Cytokinesis, karyokinesis, diakinesis. So what will be the word kinesis means in biology?
Division.
Yes,
Division.
something dividing, something getting separated, right? So here what is dividing is the chiasmata. Okay, it's called in one line, it is the termination or terminalization of chiasmata, ending of chiasmata. Okay, the chiasmata was visible in diploide, so terminalization of chiasmata. Okay, is this clear? And with diakinesis, prophase 1 ends and metaphase 1 begins. Yes or no?
Yes.
And what used to happen in metaphase 1 of mitosis? The same thing will happen, not metaphase 1 nahi. What used to happen at metaphase of mitosis? in the beginning of metaphase, the nuclear membrane will disappear, the envelope will break, and everything, all the chromosomes will come out, right, in the cytoplasm, so that they can align on the metaphase plate. But mark here that in case of metaphase 1, there are 2 homologous chromosomes carrying 4 chromatids, 2 sister chromatid each, which is aligning at the plate. This is how it is different from the mitosis. You understood?
So there would be 2 metaphasic plate formed, right?
There will be one only. Like if you, yes. So like if you're talking about lines, like yes,
Yeah,
the plate will be one, but the lines of chromosomes will be 2, because there are homologous chromosomes. For chromosome number 1, there will be 2 chromosomes carrying 2 sister chromatids. So total 4 chromatids are lined. So 2 lines will be there. And in the 2 lines also, there will be 2 chromatids in each line. Okay, you can visualize it, right? Both of you, Aisha and Iram, is it getting easier for you or not? Yeah. Okay. So let me then I ask you to make notes, because Aisha, today you have to go back and then make notes. Okay. You have to revisit, revise, and make notes. So Iram, then what happens in metaphase 1 according to your notes? Tell me. Metaphase 1 starts. So what happens in metaphase 1?
chromosomes are attached to spindle via kinetochore.
Chromosomes are attached to spindles. The same thing. Spindles
Yeah,
will
same thing.
come and start attaching, right? And
Yeah.
then they will start pulling it. So when they will start pulling it apart, that will be anaphase that begins, right? Remember, in mitosis, at the metaphase plate, there was only one chromatid of each, sister chromatids of each chromosome aligned. Yes. So the sister chromatids were being pulled apart. Here there are 2 lines now. Homologous chromosomes carrying sister chromatids are aligned. Yes. Yes.
Yes.
Good. Then anaphase 1 is same. It starts pulling apart. And then telophase 1 is when they reached at 2 different ends. So this is clear, this is clear, this is clear. Telophase is when they reach at 2 different poles and nuclear membrane starts reappearing. Okay, let me allow Iram to teach. Yes, Iram, tell telophase 1.
Homologous chromosome separates while sister chromatid remains associated at the centromere. Sorry, sir, you asked telophase?
Yes, I asked telophase.
Chromatin material decondenses, nuclear
Yes,
membrane, nucleolus, cell organelle reappears.
exactly. And you told me that there is cytokinesis 1. So this is one thing where it needs a little bit of correction. We do not call it cytokinesis because we believe, because meiosis has yet not completed. So instead of the term cytokinesis, the other term is used. Do you know what term is used? Between meiosis 1 and
Interkinesis.
meiosis 2? Yes. It is called interkinesis. The word inter means inter and intra. We learned till this.
Between.
Interschool. Yes. And kinesis means division, right? So between the actual divisions, this is between division. So between M1 and M2, the division that happens of the membrane is called interkinesis. Okay. Will you remember this? And what happens in interkinesis? 2 cells are formed, and then both the cells immediately from meiosis 1, they enter into meiosis 2. And the same thing happens in meiosis 2. But meiosis 2 is just mitosis, so it is very simple. It's exactly the same thing as mitosis. So there will be a prophase 2, but this time the prophase 2 will not have such elaborate leptotene, zygotine, pachytene, diplotin, right? Because there is no crossing over happening again. It has happened.
And so
So
DNA also won't replicate, right?
Yes. So no DNA replication here. No DNA replication. No crossing over. It only happens once. So what happens in prophase according to your notes, Iram? This is what does not happen. I told you. What happens according to your notes?
Chromatin material starts condensing, nuclear membrane, nucleolar cell organelles disappear.
Yes, the same thing is repeating. Same story of mitosis repeating. Yes. The chromosome becomes compact, nuclear membrane starts to dissolve, and in the cytoplasm, both the centrosome starts coming at the opposite poles. Yes, just like mitosis. That's why we say that it mimics mitosis. Then comes metaphase 2. What happens in this?
Same thing as mitosis.
Yes. Chromosome aligns at the equator, microtubules from opposite poles make the spindles, they come and attach to the kinetocores. But this time in meiosis-2, they are just like mitosis attaching to the sister chromatids. They are now pulling apart sister chromatids. In metaphase 1, what was being pulled apart? Homologous chromosomes. Basically, the non-sister chromatids were being pulled apart, right? Here, from the same chromosome, you are pulling apart two sister chromatids. You understand?
Yes.
Yes. Good. Then third is anaphase 2. Tell me, Iram.
Centromere splits.
Yes.
And sister chromatid move towards opposite poles of the cell.
Yes, right. And it will have the same kind of, where the centromere is lagging and the chromosomal arms are, the centromere is leading and the chromosomal arms are lagging, right? We studied in meiosis. Remember lagging and leading structure, this kind of a structure when it was being pulled to one side. This is one arm of the chromosome. This is another arm of the chromosome. Both are lagging and the centromere is leading. Forgot kya? No problem. Let me. Okay, remember. Good. Then finally comes your telophase 2. Yes, Iram?
Decondensed chromatin, reappearance of nuclear membrane, nucleolus, and cell organelles.
Yes. And finally, at the end, what will happen? Now, this time we call it cytokinesis. And it will have, so this is happening in 2 cells. So from 2 cells, finally, there will be 4 haploid cells. Yes?
Yes.
Very good. Now let me use a image that explains, summarizes everything. And I will give you some time to analyze that image on your own. I think the picture is on my downloads. Yes, this is the picture. Can you see this picture that I have pulled out? Let me make it big for you. See it. Can you read it now? Can you both read it, or shall I make it even more bigger? I'll send this picture on the group. Okay. Now, what about now? How about now?
Yes, Lord.
Is it readable now?
Yes.
Okay. So please read. And when you need me to scroll, I will scroll down. Start from the top. The first, how you have to read is interphase, S phase, see the figure, see the markings, then come to this outcome. Then go to prophase 1, see the diagram, see the markings, come to the outcome. And read it completely. So I'll ask Iram to read S phase, prophase, and Meiosis-1, and then Meiosis-2 will be read by Aisha. Okay, let's start.
S phase. Chromosomes are duplicated during interphase. The resulting sister chromatid are held together at the centromere. The centromeres are also duplicated.
Centrosomes.
Sorry, centrosomes.
Centromeres are present on chromosomes. Centrosomes are present in the cytoplasm.
Yes.
If you make this mistake, you are
also duplicated.
Yeah, and also at the figure. So you read, I will explain the figure. So can you see that in the figure, the nuclear envelope is there, but centrosomes are also there, and they will start moving towards, nothing is happening, no movement yet, because it is S phase, right?
Yes.
Just duplication happens of the DNA and of the centrosomes. Now come to prophase 1.
Chromosomes condenses and the nuclear envelope fragments. Homologous chromosomes bind firmly together along their length, forming a tetrad. Chiasmata form between non-sister chromatids. Crossing over occurs at the chiasmata. Spindle fibers emerge from the centrosomes.
Yes. Did you understand, Aisha? Can you see everything written here through the image? That spindle are moving towards opposite poles now. One is going this side, one is going other side. Nuclear envelope is fragmented, so we see these fragments, and then all the chromosomes are out in the open. Homologous chromosomes bind together along their length, so they kind of hug together. They both are hugging each other because they have to together reach the metaphase plate. So first, the homologous chromosome will get separated in metaphase 1. Then in metaphase 2, the sister chromatids of each chromosome will get separated. You understand? This is the difference. You understand, Iram, Aisha?
Yes.
Now move ahead.
Prometaphase. Homologous chromosomes are attached to spindle microtubules at the fused kinetochore shared by the sister chromatids. Chromosomes continue to condense, and the nuclear envelope completely disappears.
Yes, prometaphase is not a like actual phase. It is just combination of prophase is ending and metaphase is beginning. So it is just this and this in between, intermediate. Just to explain you, so that they all start coming towards the central plate. And then what happens in metaphase?
Homologous chromosomes randomly assemble at the metaphase plate, where they have been manued into place by the microtubules.
Yes, this word, very heavy word, maneuvered, simply means placed or put into place. Okay. Yes. So this is simple. Anaphase.
Anaphase 1, spindle microtubules pull the homologous chromosomes apart. The sister chromatids are still attached to the centromere.
Yes. So can you see now? Look at the diagram. What is getting pulled apart towards this side and towards this side is still a chromosome carrying two chromatids. So these two homologous chromosomes are being pulled apart, but still they have two chromatids. But in mitosis, this was not the case. In mitosis, there was only one chromosome at the plate, and two sister chromatids were being pulled apart right in the first metaphase, right? Because there was only one metaphase there. Here, first the two chromosomes get separated, which are homologous chromosomes. Then when they reach the plate in the meiosis-2, metaphase 2, then the sister chromatids get separated. I hope you do not have any confusion in sister chromatids, non-sister chromatids, homologous chromosomes, etc. now. Is there any confusion, Iram and Aisha?
No.
No, good. Yes, let's come to the last. Telophase 1 and cytokinesis. You know that this is actually interkinesis. Yes. Read.
Sister chromatids arrive at the pole of the cell and begin to decondense. A nuclear envelope forms around each nucleus, and the cytoplasm is divided by a cleavage furrow. The result is two haploid cells. Each cell contains one duplicated copy of each homologous chromosome pair.
Yes, right. So each cell contains one duplicated copy of each homologous chromosome pair. That's it. But it is a haploid cell. So right after meiosis 1, we reach two haploid cells. Okay. Which proves that we started with a diploid cell and two haploid cells we get after meiosis 1. So meiosis 1 is reductional in nature. Clear? I will send this figure on the group. Don't worry about it. You can I'll put it as a document file so that you can download and it can have the same resolution. Okay, Aisha Nira?
Yes, sir.
It's a handy tool you should have to revise. Okay, now let's come to meiosis 2. Aisha, your turn. So from here onwards, you can see that now there are two cells. In the end, this division will make 4 cells, right? So what's happening? Aisha.
Sister chromatids condense. A new spindle.
Read the stage also, no?
begins.
Which stage? Yeah.
Prophase 2.
Yes.
Sister chromatids condense. A new spindle begins to form. The nuclear envelope starts to fragment.
Yes. Yes. Good. Then
Prometaphase 2.
this is not in your NCERT, so don't worry about it. I told you that this is a in-between stage. It is actually a stage studied in higher classes. If you go to pursuing research in cell biology, you will study this in detail. But don't worry about it here. Yes, read.
The nuclear envelope disappears and the spindle fibers engage the individual kinetochores on the sister chromatids.
Yes. Make sense to both of you?
Yes.
Okay, good. Then metaphase 2.
Metaphase 2. Sister chromatids line up at the metaphase plate.
Simple. Just like mitosis. Now look here, the sister chromatids, they are always using the word sister chromatids. Sister chromatids means one chromosome, right?
Yes.
Single chromosome. That's why it is haploid. There it was homologous chromosomes, which means two chromosomes in meiosis 1, metaphase 1. Yes, continue.
Anaphase 2. Sister chromatids are pulled apart by the shortening of the kinetochore microtubules. Non-kinetochore microtubules lengthen the cell.
Yes, you don't have to worry about this part. Last.
Telophase 2 and cytokinesis. Chromosomes arrive at the poles of the cell and decondense. Nuclear envelopes surround the 4 nuclei. Cleavage furrows divide the 2 cells into 4 haploid cells.
Very good. So finally, from 2 haploid cells, we get 4 haploid cells. Right? These two will divide from the center like this, and we'll get 1, 2, 3, 4 haploid cells, each containing one chromatid. And 2 of these cells will be recombinant cells, and 2 of these cells will be parental cells. Remember, the one which will get this chromatid will be parental, and the one which will get these two will be recombinants. Yes. Make sense?
Yeah.
Yes.
Very good. So this is your overall. So here the meiosis is finished. One last question that is here, just like mitosis, we have to figure out what is the significance of meiosis. Tell me. What is the significance of meiosis? So first and the major significance and most important goal that meiosis achieves is that it is the mechanism by which we conserve the chromosome number in each species across generations, right? So write down, through meiosis, the conservation of specific chromosome number what do we call it? Ploidy, right? You can also call it ploidy, right? Whether it's haploid or diploid, N2, N3 and whatever. So through meiosis, the conservation of specific chromosome number of each species is achieved across generations. Clear, Aisha, Neeram?
Yes, sir.
What else? What is the other significance? Is there any other significance? Yes, what I elaborated you through the diagram. Variations. You were about to say this, Neeram?
Yeah.
See, this is called opportunistic behavior. Even if you were not, you can just say yeah. But I know you were about to say this. I trust my students and their instincts. Yes, no, I'm not doubting. Yes, the second thing is variability. So write down, it increases, meiosis increases the genetic variability, so the genetic variation. Genetic variability in the population. Through recombination. And in which phase of meiosis recombination happen? Pachytene of prophase 1. Remember that for your entrance, okay? And variations are very important for evolution, because variations, if there are no variations, then adaptation will not happen. Organisms will not be able to adapt to the changing environment. Understood, everyone?
Yes, sir.
Good. So we have finished this chapter as well. And after finishing 2 chapters, cell and cell cycle and cell division, I would now make you solve some entrance questions. Why, Iram, you are not happy that we have finished the chapter?
No, sir, that was by mistake.
What? I said that was by mistake. Okay. I think she's like, why have you finished the chapter so early? It just started to be fun. I started to love cell cycle and it finished. This is the thing about cell cycle. The moment you start understanding it, by then the cell is divided. It's done. Okay. Yes. So we have finished cell, the fundamental aspects of it, and also the cell division and cell cycle. Now I'll make you solve some actual examination NEET questions which have come in the previous years. Okay. So we'll spend rest half an hour of the class solving previous year's questions from the NEET paper. So before that, take 3 minutes. Will 3 minutes be enough to revise everything? Both the 2 chapters? How much time do you need? 4 minutes? You just have to go in a rapid mode. Just revise whatever you have. Just take a look. Okay? Yes, and your time starts now. I'll be back. I'll unmute in 4 minutes. By the time, let me pull out the questions that we will be discussing. and I'll tell you the rules after that. Okay, so just go through the notes. You can also ask if you have any doubts, but while solving the question, you are not allowed to ask doubts. If you solve it right or wrong, then we will discuss. So utilize this time. Now I will be silent. Why not happy again, Iram? What happened, Iram? You're not happy with the rules? I think it was also by mistake. Okay, cool. Take care, revise.
Sir,
Yes, Aisha.
why is DNA called the blueprint of life?
Yes, good question. See, DNA contains all the genes, right? All the genes that give that control all our characters. For example, you have a skin tone, a skin color, right? You also have a texture to your hair. You also have a color to your hair. You have a shape to your ear lobes, to the nose, to the chin, and everything is a character, right? These all these characters are defined by genes. So all that information of how you look and how your organs inside the body are functioning, all that information is present in the DNA in the form of genes. That's why it's called the blueprint. Blueprint means, have you seen those architects maps which contains every minute detail of the building? That's called the blueprint of a building, blueprint of a floor. You understood? A detailed information. So if you read that map, it is different from the map that we make of the cities or the country. It's not detailed, it's just to give you a context or idea. But a detailed blueprint map tells you about each and every corner, what millimeter, centimeter, or meters of distance will be between two things. So similarly, the blueprint is the DNA for every life, every living system, because it contains all the information of the appearance and the anatomy and the structural organization, the organ formation, everything. You understood, Aisha? That's why it's called the blueprint.
Yes, sir.
Okay, very well. Done. I hope you are starting from the first. Chapter 8, right? Which chapter you are starting, both of you, Aisha, Iram? Chapter 8 or 10?
10.
Chapter 8.
Wow, great. One is starting from 8, the other 10. Good. See, this is genetic variation right there in the class. Revisional variation. Okay, complete both. Quick.
So I have a question.
Yes, Iram.
Is steroid a type of lipid?
Yes. Steroids. Where have you heard the term steroid?
So in those assignments uploaded in recent, there.
Yes, right. Yeah. So steroid is actually they can be of two types. They can be synthetic or they can be natural. Okay. If they are natural, there are many steroidal hormones, for example, in our body, which are naturally produced. Okay. And the basis of steroid is, yes, it is kind of a lipid chain. You understand?
Yeah.
And steroid hormones, for example, the hormones, steroid hormones basically help in controlling metabolism and also your salt and water balance. So if you want to get some examples, testosterone is a steroidal hormone in males and estrogen or progesterone is a steroidal hormone in females, and these are natural ones. And they are not protein hormones, they are steroid hormones. Okay, so they are kind of different kind of lipids. They come under the lipid category. You're right. Does it answer your question, Hiram?
And one more question.
Yeah.
So if we remove the cell wall from a plant cell and place it into a drop of water, what will happen?
Yes, good question. Osmosis will happen. You have studied osmosis in class 10th? Osmosis is water moves from its higher concentration to its lower concentration. Now, if you place a cell in pure water, inside the cell there is cytoplasm, right? Cytoplasm is not pure water. Anything mixed with water makes it unpure and its concentration decreases. So in the cytoplasm, let's say 90% is water, but 10% is something else like organelles, other dissolved proteins, ribosomes, RNA, etc., etc. But in the glass, it is 100% water. So where is the concentration of water more? In the glass or inside the cell? Hiram?
Cell?
No, cell is 90% water, 10% are other things dissolved in it. Cytoplasm is just 90% water with 10% other things dissolved in it. It is a solution. But when you place that cell in a glass of water, it is pure water. You have not dissolved anything in that water, right? So where is the concentration of water more? Cell or glass?
Glass.
Yes, glass is pure water. Pure water is the highest concentration, 100% water. So water will start moving from the glass into the cell. Every cell starts taking up water. That is the reason. Have you ever washed your clothes, or have you ever, played in water or in water parks for very long?
Yeah. Long back.
Have you noticed what happens to your hands and your soft tissues? They kind of swell up.
Yes, yes.
Yes. Why? Water is like they absorb water from surroundings. Our cells absorb water. So similar thing will happen if you place a cell. Have you done this experiment where you put raisins in a glass of water and leave it overnight? What happens to the raisin?
Will swell.
It swell up, right? Because water moves from the glass into the cell where the concentration is low. It will keep moving into the cell till that point both the concentrations are equal and cell will swell up. So if you remove, and cell wall of the plant prevents this from happening. It does not allow water. It gives support to the cell. So if there is a cell wall, the cell can only swell up to the area that is the area of the cell wall. Once the cell membrane touches the cell wall, it stops swelling because cell wall is rigid like a concrete wall. That's why if you remove the cell wall, then you place a plant cell in a water, it will behave like an animal cell and it will keep swelling to a level where if it does not stop, it will burst. Does that answer your question? No?
Yeah.
Good. Okay, so is this done? Revision is done? If not, then still let's take some questions. We may not be able to complete both the chapters. So let me start with chapter 8, basics. And in the next class, before we begin the new chapter, which is biomolecules, we will do more questions, okay? Because these ultimately we have to check how much we have understood with respect to the actual question paper. So can you see this screen that I'm sharing with both of you?
Yes, sir.
Yes, so these is previous year's questions which have been asked in NEET from chapter 8. Let me start with the easier one. First question, NEET 2017, it was asked. How about Iram, you read it. Let's start. You read and read the options. Don't give me the answer. Write the answer in the chat box so that Aisha can also write separately.
Okay.
And write just to me, not to everyone, okay? Just to me. Again, to avoid cheating. Okay, read, Iram, the first.
Which of the following cell organelles is responsible for extracting energy from the carbohydrates to form ATP? Options are ribosomes, chloroplast, mitochondria, lysosome.
Yes. You don't have to think much about it. Whenever they talk about ATP, they're talking about energy and you know who produces energy. So this is just a different language in which they ask questions. Fast. You can just write A, B, C, or D options. You don't have to write full, okay? You know, good. Correct. Aisha, what is your answer? Option A, B, C, or D?
Sir, option A.
But why? Read the question again. Which of the following cell organelles is responsible for extracting energy from...
Lysosome.
Read the question once more. What is ATP? Tell me that, Aisha. What is ATP? ATP is the energy currency of the cell, right? Adenosine triphosphate. It is the energy molecule of the cell. The question is simply asking who makes ATP? Who makes energy in the cell?
Mitochondria.
Exactly. Simply it is ribosomes make proteins, chloroplast do photosynthesis, lysosome are the suicidal bag of the cell. Mitochondria is the powerhouse of the cell. Remember? By powerhouse, what do
Yes.
you mean? Yes. Good. Not a good start, but good that we are doing this. Okay, next question Aisha will read. Okay, and also, so Aisha, you have to write in the chat. Can you see there is a chat option?
Yes, sir. My enter key is not working.
Okay, then you answer, okay? But then you answer after 10 seconds. Let Iram register her answer in the chat, okay?
Okay, sir.
Count to 10 and then answer. Or count to 20. Let's give 20 seconds to Iram, right? Entrance will give you 1 minute for each question. There will be 180 questions and 180 minutes. So that's why let's give 20 seconds to Iram. Yeah. Okay, read the third question, Aisha.
Select the wrong statement. Bacterial cell wall is made up of peptidoglycan. Pili and fimbriae are mainly involved in motility of bacterial cells. Bacteria lack flagellated cells. Mycoplasma is a wall-less microorganism.
Count to 20. Let Iram register her answer. Option A, B, C, or D. Okay, you don't count. When Iram will send the answer, I'll tell that, okay, then you speak. Now, Iram will not register the answer only. Iram, are you there?
Sir, yeah, I'm doing.
Yeah, okay. We are putting unnecessary pressure on Iram. I feel bad for it. Out of all the keys, your enter key is not working, Aisha. There is a law
Yes, sir.
called Murphy's law. You know about that? It says whatever can happen in the universe will happen. So it can happen that your enter key will not work. And it did happen. Yes, Iram has given the answer. Aisha, what do you think? What is the right answer? What is the right option? ABC. What were you doing? If you're thinking
C.
now, you don't have to start thinking after Iram stops thinking. Okay? Yeah. Okay, good. So
C.
your option is C. Both of you are doing this classical mistake. I knew you will do this. That's why I asked you to read this question. The question asks, select the wrong statement. You both are selecting the right one. Select the wrong statement. So the right answer is the one which is the wrong statement. Which of the following is incorrect? C is correct. D is correct. A is also correct. Bacterial cell wall is made up of peptidoglycan. This is correct. Cyanobacteria lacks flagellated cells. Correct. Cyanobacteria are not bacteria, I told you. Sorry, cyanobacteria are bacteria. They are known as blue-green algae. They are not algae, BGA. They are cyanobacteria. Mycoplasma is a wall-less microorganism. Yes, that's true. Pili and fimbriae are mainly involved in motility. This is a wrong statement. They are not mainly involved in motility. Remember, I told you that fimbriae. What are fimbriae? Flagella are involved in motility, not fimbriae. Remember, pili and flagella. So they gave you a similar looking word and you got confused. Pili and flagella are mainly involved in motility. It's a NCERT text line. Don't worry, don't be disheartened. This is what, so we will do it in the next class as well, okay? After finishing chunk of syllabus, we will spend some time here because this is why we are doing this syllabus, right? Very good. Let's go to some difficult question now. Something that takes thinking. Okay, question number 13. Just apply some concept of endomembranous system here. Question number 13, Iram. Read.
Nuclear envelope is a derivative of microtubules, rough endoplasmic reticulum, smooth endoplasmic reticulum, or membrane of Golgi complex.
What do you think is the answer, Iram? Okay, whoever will read will tell the answer. Let's do this so that you all get equal. Other one can think of the answer. If that is right, then you're doing good. If wrong, then you learn. So Iram, you read this question. What do you think?
So you said it's related with ER, right?
Endomembrane system.
Yeah.
Endomembranous system.
Okay. So I think rough endoplasmic reticulum.
Exactly. You are right, because remember I told you that rough endoplasmic reticulum is attached to the nucleus on one side. So it is basically the nuclear membrane continuing to make RER, then it makes SER, and then it attaches to the plasma membrane, right? That is the whole system. Very good. First right answer. Claps. Good. Aisha, your turn. So read question number 21. Something similar from the endomembrane system. Question 21. Sorry.
The Golgi complex plays a major role as energy transferring organelles in post-translation modification of proteins and glycosylation of lipids, in trapping the light and transforming it into chemical energy, in digesting proteins and carbohydrates.
Yes, do not get intimidated by very, very heavy terms. Okay, it's very simple. So what do you think is the right answer, Aisha?
So I have given the answer.
Yes, I know, but I'll disclose it only after Aisha has given the answer, okay? Aisha, what do you think? The question is very simple. What is the role of Golgi bodies? Do they transfer energy?
No.
No. Do they
Option B?
play a role in? Yes, exactly. Option B. They do not trap light because that is done by chloroplast. Remember?
Yes.
The chlorophyll. They do not also digest proteins because that are done by enzymes, peptides and amylase. Carbohydrates is digested by amylase and this is by peptides. So they play a role in post-translational modification of proteins and glycosylation of lipids. They make proteins and lipids. Remember, RER is associated, sorry, whatever RER and SER made, RER makes protein, SER makes lipids, and then both of them go to Golgi for their packaging and modification. Remember, so the only job of Golgi is to package and modify things.
Yes.
And Iram gave the right answer at a lightning fast speed. I think it was not just within 10 seconds. So that's great. But I'm happy that Aisha gave a right answer, thought it carefully. You are also within time. So if you take less than 1 minute for any question, it is good from entrance point of view. Iram's turn. Okay, Iram, you read. Let's go to some difficult ones. Okay, this one. We'll do it later. Yes. Question number 29, Iram.
What is true about ribosomes? The prokaryotic ribosomes are ATS, where S stands for sedimentation coefficient. B, these are composed of ribonucleic acid and proteins. C, they are found only in eukaryotic cell. D, these are self-splicing splicing introns of some RNAs.
Sometimes they give you options just to confuse you, which you have never heard of, which is option D here. Aisha knows the answer actually. But how, if I ask you, Aisha, then Iram's term will be, and you cannot type it. Okay,
B.
let Iram answer. Iram, what is the answer according to you?
B.
B. Aisha, what was your answer?
Sir, B.
Yes, B. So very good. Both of you were within 10 seconds. Very good. Claps. Now, 47. Aisha, question number 47.
Vacuole in a plant cell lacks a membrane and contains air, lacks membrane and contains water and excretory substances, is membrane bound and contains storage proteins and lipids, is membrane bound and contains water and excretory substances. Option D.
Two. Exactly. Iram, right? Hiram has already given the answer, option D. Simple. Vacuoles have a membrane called tonoplast. It is single membranous. Very good. You both are doing great now. So you had a bad start, but you have picked up pace. You people are like Virat Kohli. Yeah. So slow start, but then boom, boom. Boundaries. Vacuoles have a single membrane structure called tonoplast, and it does not contain useful things mostly. It all contains excretory substances. So if you remember these two things, it's good. Question number 49, Iram. You have to apply some brain, fluid mosaic model. Just a hint. Yes.
Keeping in view the fluid mosaic model for the structure of cell membrane, which one of the following statement is correct with respect to the movement of lipids and proteins from one lipid monolayer to other, described as flip-flop movement? While option A, while proteins can flip-flop, lipids can not. Neither lipids nor proteins can flip-flop. Both lipids and proteins can flip-flop, while lipids can rarely flip-flop, proteins cannot.
Yes. Flip-flop is just changing, like, from going from outer membrane to inner membrane. So fluid mosaic model. Okay, first you tell me the answer. What do you think is the answer? Remember what was fluid mosaic? Don't get confused by flip-flop, tiptoe, touchdown, jump up, all these terms. Just stick to your concepts.
B.
B. Okay, and Aisha, what do you think?
Option A, I guess.
A. Okay. So Iram is less wrong. Aisha is wrong. See, Iram thinks that neither lipids nor proteins can flip-flop. She's right that proteins cannot flip-flop. Proteins can only move from one place to another. Remember I told you? It is fluid mosaic, so that the proteins, there are two types of proteins, integral and embedded, right? Embedded are only on one side. Integral is which spans from the outside to the inside completely. Remember that diagram?
Yeah.
Yeah. So proteins cannot flip-flop. Do you understand? Lipids can sometimes, very rarely, they can. But you were very close to the right answer, Iram, saying that neither lipids nor proteins can flip-flop. So just write it down.
Lipids flip-flop.
Lipids can rarely flip-flop in circumstances where proteins are moving sometimes. So when proteins are moving from one place to another, they actually are like floating in the lipid pool. Okay. So lipids sometimes can change their, because if they flip-flop, there is no difference. On the outer membrane also is a lipid, on the inner membrane also is a lipid. If they change positions, it's still on the outer side is a lipid and on the inner side is a lipid. Though this is also very rare, but it happens. It is possible. Proteins, if they flip-flop, then where will an integral protein go if it flip-flops? Right? It cannot flip-flop. It can just move from one place to another, because on the outer side of the protein, there are receptor molecules, sugar molecules. If it flip-flops, that sugar molecules will come in the cytoplasm where they have no roles. You understand? Aisha and Iram.
Yes, sir.
So some logics we can only understand through questions. So if you find difficulty, just write down through fluid, just write down fluid mosaic model says that proteins, fluid mosaic model says that proteins can move or change position. Proteins can move or change position in a lipid bilayer. In a lipid bilayer. But cannot flip-flop. Lipids can sometimes flip-flop, change positions from inner membrane to outer membrane. Okay, written?
Yes, sir.
Very good. So it's Aisha's turn now. 51, Aisha, question number 51.
Select the wrong statement from the following. Both chloroplast and mitochondria have an internal compartment, the thylakoid space bounded by the thylakoid membrane. Both chloroplast and mitochondria contain DNA. The chloroplasts are generally much larger than mitochondria. Both chloroplast and mitochondria contain an inner and an outer membrane.
Yes. What do you think is the answer?
Option C.
Option C.
Yes.
And what do you think, Iram? Iram, what do you think is the answer?
E.
Yes, C. Again, Aisha, you're falling to the trap. When
I'm
you read
correct.
for, yes, you are correct. Its right answer is option A. Aisha, when you're reading the options, see, our brain is wired. Biologically, we are always trained in our school, colleges, in life to find the right option. You know, entrance use the same training of our mind against us. It is asking select the wrong statement from the following. Okay, both chloroplast and mitochondria contain an inner and outer membrane, right? The chloroplasts are generally much larger than mitochondria, right? Because their outer membrane is bigger than their inner membrane. Remember, Aisha? They will be bigger, no? Both chloroplast and mitochondria contain DNA, again, right? But both chloroplast and mitochondria have an internal compartment called the thylakoid, is wrong. Thylakoid is only present in chloroplast. In mitochondria, there is no, the internal compartment is called, what it is called? The mitochondrial matrix. Remember the lumen and the mitochondrial matrix?
Yes.
Yes? Yeah. So you know the answer, but you're getting tricked to always find the right one. So don't get tricked, okay? So 51, the right answer is A, and Iram gave the answer pretty fast. So well done. Good. Okay. So I'll take one last question for today, and in the next class, we'll again start with doing this. Okay. So will it be fine with you, Iram, if I give the last question also to Aisha? Because anyways, you can
Yeah, no
answer in the
problem.
chat. Yeah. So let's give Aisha this chance to give the right answer, and then we'll end. Which means if you do not give the right answer, Aisha, there'll be one more question that we'll do. Okay. Now don't use it for your benefit and keep giving wrong answers so that we cover all the questions. Okay, we have to sleep. Okay, so let me choose another question which involves thinking. Others are just fact based, so that you can answer any given day. Okay, question number 71, Aisha.
Which of the following ribosomes are engaged in protein synthesis in animal cells? Ribosomes which occur on nuclear membrane and ER, ribosomes of only cytosol, ribosomes of only nucleolus and cytosol, ribosomes of only mitochondria and cytosol. Option A.
Perfect, very good. Right answer given within 10 seconds. Congratulations. Good.
Thank you, sir.
They're just tricking you. Ribosomes of only cytosol is true. There are ribosomes in cytosol. They are called free ribosomes. But in animal cell, ribosomes which occur on the nuclear membrane and the ER synthesize proteins, right? Because the mRNA which is produced inside has to come through the nuclear pores. So the moment it comes out, these ribosomes is like, dhar lete hain. They just hold it and start synthesizing protein out of it. And also ER, which contains ribosome, is called RER, just attached to the nuclear membrane. So it synthesizes protein. C and D are absurd options that it does not exist. Ribosomes of nucleolus. There is no ribosome in the nucleolus. Okay.